Okay, yes. So I decided to do it live because it's more fun. And yes, first I wanted to thank you for inviting me to giving this opportunity. I hope you will enjoy my talk. And I'm going to talk today about, as Milan introduced, about explainable AI and distributed learning. That's quite interesting, both topics. And of course, in one presentation, it's quite difficult to cover all aspects. I hope that I will encourage you to explore these topics more and understand that they are really interesting and important. And I will show you some examples of the application in the certain domains. And in some projects I'm involved myself, and some, of course, that's just some novel literature or some open source projects. So I will provide also some links. But first of all, I just want to give some brief overview what we are talking about, because Milano mentioned this artificial intelligence. Also in some other presentations, probably you heard that, but I want to emphasize that most of the time when we are talking about artificial intelligence, that's mostly about deep learning. So that's various type of the neural network architectures, or even about machine learning. So basically the ultimate goal, of course, of the field is to develop some artificial intelligence. But nowadays we mostly working with deep learning and neural networks. And why they became so popular, I think that you already saw in the presentations that the deep learning taken over in many fields, especially in the computer vision, in image analysis, segmentation, or classification, because the main difference on the global level, that's machine learning, you have to describe your features or extract your parameters or measurements, you name it, yourself, and build certain model. And then let's say in this example, you can see that you have some input data, and then you have some decision tree, and it gives you the output, like is it a car or not. And with deep learning, that makes it much more easy. Then you just pull together images or your data, and the neural network can figure out which features are the most important and just give you the prediction, is it a car or not. So that sounds great for certain application, but when we are talking some biological application, that of course bring the problem that what is known as a black box, so that we can give maybe perfect prediction, perfect classification, but we don't know why this type of the neural network or this type of the input data gave such good accuracy. And of course, for some follow-up analysis, like again, so exploring the drug treatment or some functionality, if we're talking about omics data analysis, that's of course essential. And that brings me to the first part of the explainable AI or explainable deep learning or machine learning. And in the image analysis, so that's just example, let's say, of the why it's so difficult. So that this is a nice illustration when you need to classify handwritten digits, and you see that you use all your input pixels as parameters, and then you just go through the network, and it gives you prediction. But on each layer, even with such a simple example like just 2D image of the handwritten digits, it's quite difficult to understand why certain images were classified as 9 or not like 7 or 0. And in computer vision with input images, that's probably more or less solved to a certain extent. That's example of the algorithm which goes to GradCam. And here you see that the middle image is the input image to the classification problem, something similar what Milano also showed, like a cat or dog classification. And then depends on the prediction, you can make a visualization of the heat map where the network actually looked at the image or which pixels are the most important to make a prediction. And you have something similar for instance, medical image analysis, That's example of the deep learning network, where you train network to predict, in this particular example, like brain age, and then once you train it, you can use some similar to breadcrumb method for backpropagation through your network, and then you can show actually which part of the brain was the most important for this prediction. And that's nice. That's already widely used for the medical image analysis, where you use convolutional neural network. But again, if we focus on the omics analysis, that gives quite some problem, because in the omics analysis with the gene expression, methylation, or even genotype data, you can't use convolutional neural network, because the relation between your input parameters is more difficult and not so such straightforward as the pixels or voxels on the image. And to show you that it's still doable to a certain way, I want to introduce first, I think that's one of my favorite method to analyze the data for omics. That's what calls the autoencoder. So that's a type of the deep learning network which you use for dimensionality as a dimensionality reduction technique, but that type of the technique takes into account nonlinear relation between your input data. And here you see again, so this is just a simple example with 2D images of handwritten digits. And first you start with the full data size as on this image as encoder, and then you try to reduce the dimensionality as much as possible to latent space, to this code area. So let's say you have the input digits like 64 pixels by 64, and then you reduce it, for instance, just to 10 parameters in your latent space in the code part. And then from these 10 parameters, you're trying to reconstruct the input image as accurate as possible. So of course, sometimes it depends on your data, depends on the relation of your data, but in this example, for instance, with the handwritten digits, that's quite doable, and you can reduce dramatically the dimensionality. And just to give some example of the medical application, so that's the same for instance, 3D facial data, or the same as now used in some modern algorithms in Instagram or Snapchat. So in this example, again, you start with the high dimensional data with the 3D meshes, and then you reduce dimensionality, and then you reconstruct the face. So why it's interesting? It's not about only how much you can reduce your data, but how you can use this latent space. And in this example, for instance, with the face analysis, you can explore these latent features and relate them to various outcomes. So for instance, in this example, you can link them to gender, BMI, weight, height, and you can manipulate these latent features and see how your input parameters are changing. So, and then you can relate this to the, again, so health conditions or the outcomes you're interested in. So this is just example with general characteristics, but of course, even with this analysis, you can link into the, for instance, cognitive problems or psychiatric conditions. But coming back to the omics application, this type of the analysis is now widely used, for instance, for gene expression. Then again, you use as input your matrix of the gene expression data, you reduce dimensionality, and then again, you reconstruct original data. And then you can even use this type of the network for two types of analysis. First is denoising of your data, and second, again, exploring the latent space. In next example, it will be more clear how you can do this. So this is the same type of the encoder-decoder network, but for methylation data. And then once you train the network, so you can use this latent vector to explore the methylation pattern And it's actually in unsupervised matter. If you just project or plot this latent features, you can already see in this example, you can already distinguish between healthy and cancer patient. But you can go further, and actually by exploring the latent space with a specific algorithm, I will mention this next slide, the SHARP method. So you can see actually which CpGs are important for certain class of your healthy or cancer patient in your latent space. So that's actually quite amazing. And as I mentioned, this SHARP method that's not only used for methylation data, it's a general autoencoder. It's a general framework for many type of the machine learning and deep learning methods, which actually explain your variables in a way of their importance and how they contribute to the final prediction or classification. I would suggest to check the GitHub. This is open source project. Next example is also a novel, quite recent publication, and the other actually used data combined from the electronic health records databases to also predict outcome, but also to explain why. And as you can see in this picture, so that on the bottom part, that you can see the relevance, but also importance of the parameters. So the detail, I'm not going deep in details how they did this. That's also quite interesting. I suggest to check the original paper. And then just applying this technique, you can also explore which of the parameters are the most important for your model. But coming back to the classical methods, like for instance, like GWAS. So if we work with the genotype data, that's the main problem here. So because even if we can build certain algorithms, we want to know why the prediction works so well, and which, for instance, mutations in your DNA are relevant for such prediction or for disease. And therefore, quite recent, well, already 2 years old paper, it's like a review paper about actually how we can merge together the field of omics and deep learning by building interpretable neural networks by integrating biological knowledge. And this is an example of quite recent paper where others, and I was involved in that project particular. So we just build a network, deep learning network, based on the biological knowledge. So where you build each layer based on the certain annotations, based on the gene annotation, how which SNPs belong to which genes, based on the pathway analysis, like combining genes to different pathways, or actually all these intermediate layers, they interchangeable. So you can also use information from single cell gene expression to combine genes together to specific tissues or specific cell types without even raw data. So you just have a genotype data, but your network based on the open source databases like GTEx, or nowadays there are plenty of databases of the single cell gene expression. And what is beautiful about such type network, then using the explainable algorithm, you can actually reproduce results of the normal GWAS, but also gene set enrichment analysis and other type of the post hoc analysis in one framework. So here you see that after you train network, you can see which genes are the most important for your prediction. Or if you use the data from single cell expression, you can see which cell types are the most important for prediction. And if you build your network really like deep with a lot of biological information, you can even have such type of the explainable prediction where you start with the center, so this is your prediction, and then you see the each layer is a different type of the information. Let's say some global pathways, local pathways, genes to specific diseases, etc. And you see that each layer, depending on how much this particular pathway is important. For instance, in the first layer, you see that metabolism, the green one, is the least one of the least important for this example. But within the metabolism, the lipid metabolism is the most important. So, and you can explore this this type of the explanation for your network basically without any post-hoc analysis. So you just train your algorithm and it's ready for interpretation. And of course, the same type of the network you can use biological knowledge also used for not only for SNPs, but in this example, for instance, for microbiome. So we're also based on some biological knowledge. You can combine your layers or build your layers based on the, let's say, family and some species structure. And then again, after you train network, you can use the same methods what I just showed in the previous slides for the SNPs analysis. You can also explore why your prediction works so well. But that's, of course, nice when you have all data in one place. And I think nowadays, for the omics analysis, of course, it becomes more and more feasible, especially given some open source databases or biobanks like UK Biobank. But still, would be nice if it's possible to combine data from different sources, from different universities all together. The same way as, again, most of you know probably like what is done in GWAS meta-analysis. So this is a schematic of the site type of the analysis where you have a green, it's like all sites involved in the GWAS, and then yellow is a central part, and then everyone share the summary statistics with central part, and then you perform meta-analysis. So, but this is a simplistic example of the federated learning, which is not applicable for deep learning or machine learning directly. But what we would want, for instance, is if it's possible to build the same interpretable, for instance, deep learning model, but then have, without sharing the raw data, so to preserve them in the local sites, to run the algorithms distributedly. So where you just share some parameters during the training of your algorithms between sites, and then at the end, you preserve your privacy, you don't share the data, but then at the end, you have a final result as a pooled analysis if you would train your deep learning or machine learning method on the whole data set. And just to introduce the different concepts, so again, of such methods, it's either can be with some central site involved, where still you share the model, you train the model locally, you update the model with the central site, and then you aggregate the results. But it also can be a peer-to-peer structure where you don't have centralized central site, you just share some parameters within all sites involved in your analysis and also models, and you train them all simultaneously, and at the end you aggregate results and you get your final conclusions. Of course, the topology of such type of analysis can be also quite different. So that's again, so as I said, it can be centralized, it can be decentralized, and this involves, of course, let's say how fast you want to do such type of analysis, because some type of the topology is much faster, and also because it's done through the network or internet connection, you can take this into account by building the topology for your certain analysis, because sometimes it's faster than you have decentralized, but sometimes it's faster than you have centralized architecture. And just to mention that actually this framework, they exist in general, not only for medical or omics application. So one of the most popular now is like Monai framework. It's an open source project. So based on the PyTorch, and that's of course mostly used now for medical application, but it becomes to use also for medical image application analysis, but also it becomes to be used for omics analysis. Another one is like even more general framework, SIFT, so which is also includes a lot of open source project. You can find it on GitHub as well. So that's a framework also general, and you can adapt it quite easily for your specific application in medical field. And the third one I wanted to mention, this one is from NVIDIA. It's not open source, but it's open for, so it's free to use. Again, so even this slide, you see that they mostly use for medical image analysis, but it's also quite easy adaptable for omics type of the data. And this one based on the TensorFlow backend, if it's interested. So, but you can check this, of course, also on the websites of these frameworks. And within, actually, I want to finish my presentation, and thank you for your attention, and please ask questions if you have any.